My name is Akansha and I am a PhD student at IIIT Hyderabad. I am presenting a poster titled Deciphering Metabolic Landscape of Gynecologic and Breast Cancer, a single-cell network perspective. Gynecologic and breast cancer have significant impact on women's health worldwide. One of the key challenges in improving treatment outcomes lies in the inherent heterogeneity of cancer metabolism. Gaining insights into the metabolic diversity, particularly at single-cell resolution, is important for developing more effective and personalized treatment strategy. By leveraging single-cell RNA sequencing data, we computed the metabolic pathway activities and inferred cancer-specific protein-protein interaction network and gene regulatory networks. We explored the crosstalk between these networks to identify the key alterations in metabolic regulation. In order to characterize the metabolic heterogeneity, we identified significantly over-dispersed pathways that form the basis for clustering malignant cells. We obtained 5 clusters of endometrial, cervical, ovarian, ER-positive breast cancer and 6 clusters of TNBC. The chi-square test shows the significant correlation between these clusters and patient information. Our study reveals both inter and intra-tumor metabolic heterogeneity across cancer, with significant variation in major pathways such as oxidative phosphorylation, glycolysis, cholesterol, and redox metabolism. We observed a complex interplay between oxidative phosphorylation and glycolysis, which are mutually exclusive in endometrial and ovarian cancer, while both processes are upregulated in one TNBC patient. This finding indicates the cancer cell exhibits metabolic plasticity, enabling them to switch between oxidative phosphorylation and glycolysis and occupy a hybrid state based on oxygen availability and metabolic demand. Here figure represents the metabolic landscape of TNBC and ER-positive breast cancer. Next, malignant cell-specific PPI network was used to identify metabolic modules, which uncovers connectivity pattern among metabolic genes. We found two modules to be significantly enriched in metabolic pathways in endometrial cancer, ovarian cancer, and TNBC, while we found three modules for cervical and ER-positive breast cancer. For endometrial cancer, module 6 is enriched in lipid metabolism, while module M8 is enriched in oxidative phosphorylation. This observation is consistent with pathway level analysis. Similar results were obtained for ovarian cancer. The module M5 of ER-positive breast cancer is enriched in glycine, serine, and threonine metabolism, and alanine, aspartate, and glutamate metabolism. It has also similar enrichment profile to the module M6 of endometrial cancer. The crosstalk between cancer-specific GRN and PPI networks reveal co-regulatory network which highlight disrupted metabolic processes along with their key transcriptional regulators. Transcription factor related to ER stress and immune response and cell proliferation along with HIFs and SERBFs were found to drive metabolic reprogramming in gynecological and breast cancer. In TNBC, SERBF2 is associated with module M10, targeting critical genes of cholesterol and fatty acid metabolism. The comprehensive analysis of single-cell RNA sequencing data reveals heterogeneity in metabolic processes among malignant cells and uncovers the underlying regulatory mechanism driving metabolic dysregulation in gynecologic and breast cancer. This pipeline can also be applied to decipher the metabolic regulation of any disease at single-cell resolution. Our study provides a foundation for further investigation into the pathophysiology of cancer metabolism and developing targeted therapies that disrupts metabolic pathways critical to cancer progression and resistance. If you have any query, feel free to ping me. Scan the QR code to get more details about this work. Thank you.